Welcome to The Explainer. Today, we're diving into a discovery that's literally rewriting the rulebook on biology, changing how we see proteins from fragile little creations to robust, programmable machines. So, for decades, this was the central idea in biology. Proteins, the little workhorses doing everything in our cells, were thought to be incredibly delicate. But a brand new, groundbreaking study is flipping everything we thought we knew on its head. Okay, so to really get why this is such a huge deal, we've got to first understand the old idea it's replacing. A belief that pretty much shaped biology for generations. Yeah, the traditional picture was that proteins were like a super complex jigsaw puzzle, or maybe even better, a house of cards. The thinking was their core was so tightly packed, so precisely put together, that even the smallest change, one wrong amino acid, could set off a catastrophic domino effect, and the whole structure would just collapse. And you can kind of see why they thought this, right? The numbers involved are just mind-bending. A relatively short protein, just 100 amino acids long, has more possible sequences, that's 20 to the power of 100, than there are atoms in the entire known universe. Seriously. So it felt like evolution must have been walking on this impossibly narrow tightrope to find the very few sequences that actually worked. And this belief, well, it created a massive bottleneck in the real world. Thinking proteins were so fragile meant progress was just painfully slow. Scientists had to be so careful making these tiny, cautious tweaks because they were terrified of breaking the entire machine. This turned designing new drugs or enzymes into this high-risk, super expensive game of trial and error. But what if that whole idea was just wrong? A new discovery is now painting a completely different and, frankly, way more exciting picture. And this is where our researchers enter the story. Escobedo, Four, and Lenner. Their team, a collaboration between the Center for Genomic Regulation and the Wellcome Sanger Institute, decided to put this old house of cards idea to the ultimate test, at a scale nobody had ever even dreamed of before. And this table here, it just lays out the shift perfectly. We're not talking about a house of cards anymore. We're talking about something more like Lego bricks. Instead of being super fragile, their cores are amazingly robust. And where we once thought a single mutation was a total disaster, we now know that only a few of those load-bearing amino acids are really critical. The whole system is just so much more forgiving than we ever imagined. So how'd they pull this off? They used this really ingenious three-step process. First, they created enormous libraries of over 500,000 different protein versions with randomized parts. Then, using this incredible technique called deep mutational scanning, they tested the stability of all of them all at once inside actual living cells. And finally, they used machine learning to sift through this mountain of data to find the simple, hidden rules that were going on. Okay, so that brings up a really big question. If the protein structures are so tough, so robust, why don't all of these stable versions actually work? Well, this leads us to the real key to the whole story, a hidden evolutionary rule that explains everything. The answer is a phenomenon called a lustery. Just think of it like this. You press a button on one side of a remote, and it makes something happen on the other side, even though those two parts aren't physically touching. It's communication across a distance. That's a lustery. And this is exactly what the study found. A protein could have a perfectly solid structure, the Lego castle is built, it's not going to fall over, but a change in the core could send an allosteric signal that breaks the protein's function somewhere else entirely. It's like it secretly flips the off switch from far away. And this one insight, it solves the great paradox. It means that proteins are structurally tough, like Lego, which gives evolution a huge, forgiving playground to work in. But their function is finely tuned and limited by these long-distance allosteric communications. And that's why only some combinations are ultimately successful. But discovering this new rule wasn't even the end of the story. It gave the researchers what you could almost call a new scientific superpower. Because the rules turned out to be so much simpler than anyone guessed, they could actually be captured in straightforward energy models. The team found out that protein stability doesn't depend on a bunch of complex, unknowable interactions, but on simple additive rules you can actually calculate.
But, okay, so they built these new models. How well did they really work? Well, the ultimate test was to see if their predictions held up against nature itself, against proteins that have been evolving for eons.
The results were absolutely staggering. A model that was trained on a single human protein could accurately predict the stability of related proteins from all across the tree of life. We're talking everything from bacteria to plants to insects that have been evolving separately for over 1 billion years. The fundamental rules are just that universal. So, what does this all mean for us? This fundamental discovery isn't just for textbooks, it has real-world applications that could completely change our lives. This new understanding is a total game changer. We can finally move past the slow, painstaking trial and error and start doing rapid, bold, and reliable protein design. This means we can develop more stable vaccines and medicines faster, we can engineer more efficient enzymes for green industries, and we can even get a much deeper understanding of thousands of genetic diseases. As the senior author Ben Lenner says, we're moving toward a future of programmable predictive biology, where we can accurately figure out the effects of a DNA change on a computer before we ever even have to step into a lab. By showing us the simple, robust, Lego-like rules that life has been using all this time, this research does so much more than just update a textbook. It's like we've been handed a brand new engineering manual for biology itself. So the question is no longer if we can design life, but what we should choose to build.